Hey everyone, and welcome. If you're heading into the world of pharmacy, you're going to want to know about this. Today, we're talking about a class of drugs you will absolutely be dealing with in your careers, bispecific antibodies. So let's break down exactly what you need to know to be ready for the clinic. All right, so let's get right to the core of it. What's the main difference here? Well, for a long time, we've had monoclonal antibodies. You'll see them called MBAS. Think of them like a simple key that fits one specific lock. It binds to one target, and that's it. But bispecific antibodies, or BABs, are the next level. They're engineered to have two totally different arms, which means they can grab onto two different targets at the very same time. And that right there is the game changer. That's what makes them so incredibly powerful. So, here's our roadmap for today. First, we'll talk about why we're moving beyond traditional monoclonal antibodies. Then we'll get into the nitty-gritty of how they work. I call it the two-hook mechanism. After that, we'll look at some actual drugs you'll see in the clinic. And we'll wrap up with the most important part for you, what your core role as a pharmacist is in all this. Okay, so we're talking about moving from that single key to something more like a master key, right? A bispecific antibody doesn't just open one door. It can engage two different things, two different proteins or cells at the same time. And by doing that, it unlocks totally new functions. This isn't just a small step forward, it really opens up a whole new playbook for how we can treat diseases. So, how does this actually work down at the cellular level? What's the mechanism? The best way to think about it is to picture a bridge. A bispecific antibody is basically an engineered connector. And what it connects, well, that's what makes this whole thing so revolutionary, particularly in the world of cancer treatment. So here's the most common way we see this bridge concept in action. The antibody literally grabs one of the patient's own T cells, the super powerful soldiers of our immune system, and it physically drags it over and connects it directly to a tumor cell. The antibody is like a microscopic tether, forcing an introduction between the killer cell and its target. It's basically saying, hey, T cell, meet the cancer cell I need you to destroy. And listen, if you need a super simple way to remember this for your exams or, for real life, just think of it like this. Bispecific equals two hooks. That's it. It's got one hook for one target and a second hook for another. These hooks create this connection that just wouldn't happen on its own. Simple as that. All right, let's do a quick play-by-play of how these T-cell engagers work. It's really a four-step process. Step one, the first arm of the antibody grabs onto a protein on the surface of the tumor cell, say CD19. Step two, the second arm then latches onto a protein on a T-cell, usually CD3. Now step three, this forced connection immediately activates the T cell. It's like flipping a switch. And finally, step four, that activated T cell goes to work, releasing all these toxic granules that just obliterate the tumor cell. We call this redirected lysis, which is just a fancy way of saying you are literally redirecting the patient's own immune system to go kill the cancer. Pretty cool, right?